Have you ever wondered what makes us so unique? What are the mechanisms underlying growth and development? How resistance to adverse environmental factors is regulated? And how do the species evolve? Why DNA the molecule of life? And is the genetic code so universal? Through the history of human civilizations, molecular biology holds hidden gems of inquiry. For instance, let's look at the viruses. All of us know that viruses cause millions of deaths. This year, COVID-19 pandemic was yet another proof of how devastating they can be. But are the viruses force only? Actually, some of them can be beneficial for our health and even save us from other diseases. Being the part of our genome, when speaking about the endogenous retroviruses, they influenced our origin as species. Molecular-related techniques influence a wide range of studies, including the creation of transgenic animals, new plant varieties, functional gene and others, study of disease models and mechanisms. All of this and a bit more, making molecular biology the science of the 21st century, is a new course, Methods of Molecular Biology, presented by Al-Farabi Kazakh National University with the kind support of Peter the Great, St. Petersburg Polytechnic University. My name is Dr. Ayjan Jusupova, but friends call me AJ, and I will be your instructor for this course. Initially rising at the intersection of such disciplines as biochemistry, genetics, and physics, molecular biology was able to lay the foundation for many interesting achievements of modern engineering mind, which brought an invaluable contribution to the development of medicine, agriculture, and other latest technologies. After completing this course, you will see the interaction of molecular biology with modern achievements in related fields of science and understand the specific features of structure and molecular biological processes, as well as principal research techniques of molecular biology. The course is planned as 5 basic modules. From the first module, you will learn about the genetic diversity of viruses, specifics of virus-host interactions, prevention, and treatment of viral diseases, and a bit about viruses used in biological research. Second module addresses such questions as microbial diversity, characterization of bacterial operons and gene expression, molecular mechanisms of plant-bacteria interactions, bacterial pathogenicity, as well as such techniques as molecular cloning and transformation. Third module is devoted to such topics as plant diversity, mechanisms of plant signal transduction, methods of plant biotechnology and plant breeding, as well as of their molecular aspects. From the fourth module, you will learn a few interesting details on animal diversity and behavioral biology, animal biotechnology, its methods, as well as of their molecular aspects. The last, but not the least, Module 5, discusses the golden standard of the molecular biology, polymerase chain reaction, as well as technique without which genome projects would most likely be possible. Sequencing. Another emphasis will be given to the classification and application of molecular markers. And of course, we could not omit the pharmacogenomics and gene therapy. Each context lecture is presented in the form of a video, accompanied by the text for additional reading, glossary, references, as well as 10 questions of non-graded test. In the course, you will also find 5 graded tests. In the end of the course, there will be single individual assignment for self-assessment as your final exam. Education is a passport to the future, for tomorrow belongs to those who prepare for it today. Please, feel free to leave your questions and comments in the forum, and we'll be happy to answer them. I hope you enjoy the course. Looking forward to seeing you soon. AJ. It's my honor and privilege to welcome you to the first lesson of the new course entitled Methods of Molecular Biology. Today, we are going to start with the first lecture of the Module 1, Molecular Biology of Viruses, and we will discuss viral structures and types of genetic material in them. But first, let's start with the definition. What is a virus? Viruses are not cells. They are obligate intracellular parasites and can replicate only within a host cell. Actually, a virus is a very small infectious particle consisting of nucleic acid and a protein coat which protects it. We understand that viruses are tiny structures, but have you ever thought how tiny are they? Viruses are usually much smaller than bacteria, with the vast majority being submicroscopic, and definitely they could never be seen by a naked eye. In terms of their absolute numbers, viruses appear to be the most abundant biological entities on planet Earth. The best current estimate is that there are 10 in 31 virus particles in the biosphere, which corresponds to roughly 10 innate viruses to match every cell in our bodies. The assertion about the total number of viruses is supported by measurements using both electron and fluorescence microscopy. Viruses were detected indirectly long before they were actually seen. The history of virology, the scientific study of viruses, and infections they cause, began in the closing years of the 19th century. Although Louis Pasteur and Edward Jenner developed the first vaccines to protect against viral infections, they did not know that viruses existed. The first evidence of the existence of viruses came from experiments with filters that had pores small enough to retain bacteria. In 1892, Dmitry Ivanovsky used one of these filters to show that sap from a diseased tobacco plant remained infectious to healthy tobacco plants despite having been filtered. Martinus Beijerinck called the filtered infectious substance a virus, and this discovery is considered to be the beginning of virology. The origins of molecular biology lie in early studies of viruses that infect bacteria, so-called bacteriophages, which played a central role in the discovery of the genetic role of nucleic acids. The amount and arrangement of proteins and nucleic acid of viruses determine their size and shape. The nucleic acid and proteins of each class of viruses assemble themselves into a structure called a nuclear protein or nucleocapsid. Some viruses have more than one layer of protein surrounding the nucleic acid. Still others have a lipoprotein membrane called an enveloped, derived from the membrane of the host cell that surrounds the nucleocapsid core. Penetrating the membrane are additional proteins that determine the specificity of the virus to host cells. Capsid proteins interact by highly specific, flexible, non-covalent contacts. Long terminal extensions and loops make each viral capsid unique. Virus capsids are essentially molecular containers. What do they contain? What are the charge of their contents? Viral genomes may consist of either double or single-stranded DNA or double or single-stranded RNA. Depending on its type of nucleic acid, a virus is called a DNA virus or an RNA virus. As we know, DNA or deoxyribonucleic acid is a blueprint of life, coding genes responsible for certain traits. Its double helix structure was established by James Watson and Francis Crick based on images produced by Rosalind Franklin in 1953. It is made of repeating subunits called nucleotides, which consist of phosphate, deoxyribose sugar, adenine, thymine, guanine, and cytosine bases connected by the hydrogen bonds. RNA instead is single-stranded, contains ribose and uracil instead of thymine. The protein and nucleic acid constituents have properties unique for each class of virus. When assembled, they determine the size and shape of the virus for that specific class. Here, we can see basic division of DNA and RNA-containing viruses on enveloped and non-enveloped types. Non-enveloped viruses are composed only of capsid protein and nucleic acid. Non-enveloped viruses such as rotavirus, adenovirus, and hepatitis A virus are more resistant than enveloped viruses like influenza virus and are not inactivated by agents affecting lipids such as alcohols, ether, cationic surfactants, and chlorhexidine. As we know, some human diseases are caused by viruses. For instance, cervical carcinoma is caused by human papillomavirus types 16, 18, 31. Possible causes of diarrhea are Norwalk virus and rotaviruses. There are 4 types of influenza viruses: A, B, C, and D. Human influenza A and B viruses cause seasonal epidemics of disease almost every winter. The emergence of a new and very different influenza A virus to infect people can cause an influenza pandemic. Influenza type C infections generally cause a mild respiratory illness and are not thought to cause epidemics. Influenza D viruses primarily affect cattle and are not known to infect or cause illness in people. As we could see today, viruses stimulate some great ideas in a structure, dynamics, function, complex. In particular, understanding the complex function of a system by considering its components, the possibility of creating complex forms in biological systems through self-assembly, using a simplified model of the system to understand a wide range of more complex phenomena. Please, feel free to leave your questions and comments in the forum, and we'll be happy to answer them. Good luck, and looking forward to seeing you soon. AJ. Today, viruses are so small and simple they cannot even replicate on their own. Viruses carry only the essential genetic information they need to be able to sleep inside a host cell and win over in, making new copies of the virus. The influenza virus, for instance, has only 14 protein coding genes. Because viruses are usually so basic, many biologists didn't think they could even be classified as a life form. But just over a decade ago, our view of viruses began to shift. French scientists who were examining a mystery microbe that looked like a bacterium but was genetically quite different to bacteria realized they discovered a giant virus. They named this bacteria lookalike the mimivirus. And it was not only physically large. It carried more than 1,000 genes, which was only a few hundred genes smaller than in some bacteria. Several giant viruses have been discovered since, with Pandora viruses packing around 1,100 genes. For the more abstract question of whether viruses qualify as life, Katana Knowles argues that if viruses are descended from living cells, they are still alive now, but in a very unique way. When viruses infect a cell, that reunion forms a complete living system. So many different types of viruses exist that nearly every living organism has its own set of viruses that try to infect its cells. Even the smallest and simplest of cells, prokaryotic bacteria, may be attacked by specific types of viruses. To infect a host cell, the virus must first inject its own nucleic acid into the cell through the plasma membrane and, if present, the cell wall. The virus does so by either attaching to a receptor on the cell surface or by simple mechanical force. The virus then releases its genetic material, either single or double-stranded RNA or DNA, into the cell, and cell becomes infected. The virus nucleic acid uses the host cell metabolic machinery to make large amounts of viral components. In the case of DNA viruses, the DNA transcribes itself into messenger RNA molecules that are then used to direct the cell's ribosomes. One of the first polypeptides to be translated destroyed the host DNA. In retroviruses, which inject a RNA strand, a unique enzyme called reverse transcriptase transcribes the viral RNA into DNA, which is then transcribed again into RNA. Once the viral DNA has taken control, it uses the host cell's machinery to synthesize viral DNA, protein, and start multiplying. About 25 minutes after the initial injection, approximately 200 new single individual particles of a virus, so-called virions, are formed. Once enough virions have matured and accumulated, specialized viral proteins are used to dissolve the bacterial cell wall. The cell bursts. It undergoes lysis due to the high internal osmotic pressure, water pressure, that can no longer be constrained by the cell wall. This releases progeny virions into the surrounding environment, where they can go on to infect other cells, and another lytic cycle begins. The phage that causes lysis of the host is called a lytic or virulent phages. Some lytic phages undergo a phenomenon known as lysis inhibition, where a completed phage progeny will not immediately lyse out of the cell if extracellular phage concentrations are high. In contrast, the lysogenic cycle does not result in immediate lysis of the host cell. Those phages able to undergo lysogeny are known as temperate phages. Their viral genome will integrate with host DNA and replicate along with it fairly harmlessly, or may even become established as a plasmid. The virus remains dormant until the host conditions deteriorate, perhaps due to depletion of nutrients, and then the endogenous phages known as prophages become active. At this point, They initiate the reproductive cycle, resulting in lysis of the host cell. And the lysogenic cycle allows the host cell to continue to survive and reproduce. The virus is reproduced in all of the cell's offspring. An example of a bacteriophage known to follow the lysogenic cycle and the lytic cycle is the phage lambda of Escherichia coli. Viruses that infect plant or animal cells may also undergo infections where they are not producing virions for long periods. An example is the animal herpes viruses, including herpes simplex viruses, which cause oral or genital herpes in humans. In a process called latency, these viruses can exist in nervous tissue for long periods of time without producing new virions, only to leave latency periodically and cause lesions in the skin where the virus replicates. Even though there are similarities between lysogeny and latency, the term lysogenic cycle is usually reserved to describe bacteriophages. As we could see today, coexistence of viruses where their hosts can be viewed as a molecular arms race between the virus and host elimination mechanisms. Continuous interactions between host and pathogens during their coevolution have shaped the immune system, and in turn, viruses have manipulated host immune control mechanisms to facilitate their propagation. Please, feel free to leave your questions and comments in the forum, and we'll be happy to answer them. Good luck, and looking forward to seeing you soon. AJ. Welcome to the third lecture of the Module 1, Molecular Biology of Viruses, entitled Prevention and Treatment of Viral Diseases. A special cell of the immune system called a T cell circulates looking for infections. One type of T cell is called a cytotoxic T cell because it kills cells that are infected with viruses with toxic mediators. Cytotoxic T cells have specialized proteins on their surface that help them to recognize virally infected cells. These proteins are called T cell receptors. Each cytotoxic T cell has a receptor that can specifically recognize a particular antigen peptide bound to MHC molecule, part of a special major histocompatibility complex, which is a set of genes that code for cell surface proteins essential for the acquired immune system to recognize foreign molecules. The main function of MHC molecules is to bind to antigens derived from pathogens and display them on the cell surface for recognition by the appropriate T cells. If the T cell receptor detects a peptide from a virus, it warns the T cell of an infection. The T cell releases cytotoxic factors to kill the infected cell and therefore prevent the survival of the invading virus. Viruses are highly adaptable and have developed ways to avoid detection by T cells. Some viruses stop MHC molecules from getting to the cell surface to display viral peptides. If this happens, the T cell doesn't know there is a virus inside an infected cell. Virally infected cells produce and release small proteins called interferons, which play a role in immune protection against viruses. Interferons prevent replication of viruses by directly interfering with the ability to replicate within an infected cell. They also act as signaling molecules that allow infected cells to warn nearby cells of a viral presence. This signal makes neighboring cells increase the numbers of MHC class I molecules upon their surface, so the T cells serving the area can identify and eliminate the viral infection. Viral replication triggered the host to initiate RNA interference, an ancient mechanism that chops the double-stranded RNA into 21 to 23 nucleotide-long fragments called small interfering RNA siRNA, which target and destroy the virus single-stranded genome. A simplified model for the RNA pathway is based on two steps, each involving ribonuclease enzyme. In the first step, the trigger RNA, either double-stranded or miRNA, primarily transcript, is processed into a short interfering RNA siRNA by the RNase E2 enzymes dicer and DROSHA. In the second step, siRNAs are loaded into the effector complex RNA cell primarily transcript. Two steps, each involving ribonuclease enzyme. In the first step, the trigger RNA, either double-stranded or miRNA, primarily transcript, is processed into a short interfering RNA siRNA by the RNase E2 enzymes dicer and DROSHA. In the second step, siRNAs are loaded into the effector complex RNA cell lensing complex, RISC. The siRNA is unwound during RISC assembly and the single-stranded RNA hybridizes within the mRNA target. Gene silencing is a result of nucleotic degradation of the targeted mRNA by the RNase enzyme organate slicer. If the mRNA duplex contains mismatch, the mRNA is not cleaved. Rather, gene silencing is a result of a translational inhibition. Currently, RNA interference is available research tool both in cell culture and in living organisms, because synthetic double-stranded RNA introduced into the cells can selectively and robustly induce suppression of specific genes of interest. RNA interference may be used for large-scale screens that systematically shut down each gene in the cell, which can help to identify the components necessary for a particular cellular process or an event such as cell division. The pathway is also used as a practical tool in biotechnology, medicine, and insecticides. Viruses can also be removed from the body by antibodies before they can get the chance to infect a cell. Antibodies are proteins that specifically recognize invading pathogens and stick to them. This binding serves many purposes in the eradication of the virus. Firstly, the antibodies neutralize the virus, meaning that there is no longer capable of infecting the host cell. Secondly, many antibodies can work together, causing virus particles to stick together in a process called agglutination. Agglutinated viruses make an easier target for immune cells than single particles of virus. A third mechanism used by antibodies to eradicate viruses is the activation of phagocytes. A virus-bound antibody binds to receptors called Fc receptors on the surface of phagocytic cells and triggers the mechanism known as phagocytosis, by which the cell engages and destroys the virus. Finally, antibodies can also activate the complement system, which opsonizes and promotes phagocytosis of viruses. Complement can also damage the envelope, the phospholipid bilayer, that is present on some types of viruses. 4 types of vaccines are currently available. Live virus vaccines use a weakened attenuated form of the virus. The measles, mumps, and rubbers MMR vaccine and the Varicella chickenpox vaccine are examples. Killed inactivated vaccines are made from a protein or other small pieces taken from a virus or bacteria. The whooping cough pertussis vaccine is an example. Toxoid vaccines contain a toxin or chemical made by the bacteria of virus. They make you immune by the harmful effects of the infection instead of the infection itself. Examples are diphtheria and tetanus vaccines. Biosynthetic vaccines contain man-made substances that are very similar to pieces of the virus or bacteria. The hepatitis B vaccine is an example. Sometimes protein fails because the host immune system simply does not respond adequately or at all. Lack of response commonly results from clinical factors such as diabetes, steroid use, HIV infection, or age. It also might fail for genetic reasons if the host immune system includes no strains of B cells that can generate antibodies suited to react effectively and bind into the antigens associated with the pathogen. Adjuvants commonly are used to boost immune response, particularly for older people 50 to 75 years and up, whose immune response to a simple vaccine may have weakened. Let's look on the example of influenza virus. Current vaccine programs rely heavily on the vaccine's ability to stimulate neutralizing antibody responses to the hemagglutinin protein, which plays an important role in the release of the viral RNA into the cell by causing fusion of viral and cellular membranes. One of the biggest challenges to an effective vaccination program lies on the fact that influenza viruses are ever-changing, leading to antigenic drift that results in escape from earlier immune responses. Vaccine platforms or manufacturing technologies being tested to improve vaccine efficacy are heterogeneous between different species and/or either tailored for epidemic or pandemic influenza. Antibodies against the H globular head neutralize infection by either preventing binding to cell receptors or membrane fusion. Figure A. Stock-specific antibodies do not usually neutralize the virus measured by using standard assays, but protect through inhibition of either entry, viral release by mediating antibody-dependent cell-mediating toxicity or complement cytotoxicity. Figure B. Antibodies that inhibit Rna activity interfere with the viral release from the cell surface, reducing the amount of virus progeny produced by the infected cell and may also mediate ADCC. Figure C. M2 protein is a relatively conserved transmembrane protein of influenza viruses, and its extracellular domain MTE has been extensively explored as a universal antigen candidate for vaccines. Anti-MTE antibodies do not neutralize the virus. Instead, these antibodies are thought to prevent viral budding, mediate killing of infected cells by NK cells or macrophages through ADCC, Fc opsonation, or complement activation. As we could see today, viruses are highly adaptable and scientists still seek to find ways of combating them. Please feel free to leave your questions and comments in the forum, and we'll be happy to answer them. Good luck, and looking forward to seeing you soon. AJ. It's astonishing that with our more than 35 combined years of working in the field of virology, we continue to read on a regular basis about novel, emerging viruses infecting species from all three domains of life. Modification and genetic engineering methods can be used to make modified genomes that can be carried into plants and animals by viruses acting as vectors or vehicles, thus making them more productive. In a similar fashion, viral therapy uses viruses to genetically modify disease cells and tissues for treatment of cancer. Gene therapy is the insertion of genes into a person's cells and tissues to treat a disease. In the case of a genetic disease, the defective gene is replaced with the working gene. Although the technology is still new, it has been used with some success. Scientists have focused on gene therapy for diseases caused by single gene defects such as cystic fibrosis, hemophilia, muscular dystrophy, and sickle cell anemia. An emerging field in the cancer therapy comprises alternative therapies that use viruses to kill cancer cells selectively. The idea for this approach came through early observations of cancer regression in patients suffering from unrelated viral infections. In the past 2 decades, viruses from a variety of different families, adenoviridae, hespiviridae, parvoviridae, picornaviridae, poxviridae, had been studied for their potential use as anti-cancer agents. Due to their tropism for tumors and their ability to replicate selectively in and eventually lyse cancer cells without harming non-cancerous cells, they are referred to as oncolytic viruses. Currently, multiple phase 1 to phase 3 clinical trials are ongoing for the treatment of various cancer types, including hepatocellular carcinoma, glioblastoma multiforme, colorectal cancer, and cancers of the lung, breast, prostate, pancreas, bladder, and ovaries. In contrast to oncolytic viral therapies, where the treatment is based on viral replication and cell death, non-replicating viruses are being utilized as vectors for corrected gene delivery. The goal of virus-mediated gene therapy is the delivery and expression of the therapeutic genes to desired target cells to restore the function of the defective gene for the treatment of monogenic disorders. Viral gene therapy uses the natural capacity of viral particles to protect the encapsulated nucleic acid from degradation and to deliver the DNA to the nucleus. For the ideal gene therapy vector, the viral wild-type genome is almost entirely substituted with the recombinant transgene expression cassette. This aspect is a major difference compared with the oncolytic viruses used in anti-cancer therapies, which encode many viral genes. In hundreds of ongoing clinical trials, the most commonly used vectors for gene therapy are adenoviruses, retroviruses, lentiviruses, and adeno-associated viruses. Each system has its pros and cons that must be considered prior to use to ensure efficient gene delivery and expression for clinical success. Vaccines against polio, measles, chickenpox use live and weakened viruses causing the disease or dead virus particles. These, when introduced into a healthy individual, help the immune system to recognize and mount an immunity against the virus. The body remembers the organism and attacks it in case of a later infection, thus preventing the disease. Virus-directed enzyme product therapy is a therapy when the target cells are inserted with an enzyme that can activate an inactivate precursor or inactivate form of a cytotoxic drug that is administered systematically. Thus, the active cytotoxic form of the drug is only produced where the relevant enzyme is present and active. For example, An adenovirus expressing the thymidine kinase enzyme of herpes simplex virus can be combined with the systematic administration of ganciclovir, which is converted by the TK to its active form only in cells where this enzyme is present. This is used in HIV treatment. Viruses can also be used to control damaging pests. Traditionally, this hadn't been used in agriculture, but applications exist in the control of agents important to human health as well. The types of agents used for this purpose may prey on the target species, may be parasites on the target pest, be pathogens or cause disease in the target species, or may be competing species. Viruses used for pest control are commonly pathogens causing disease of the target species. Although they account for a small amount of total pesticide use, viruses are used for the control of multiple species of insects and also for rabbits. Biological agents can produce long-lasting effects and in some cases are able to spread among the target population. They have also been recognized as inherently less toxic than conventional pesticides. Their disadvantages include limited rate of action, slow effects compared to chemical agents, high cost of initial treatment, low environmental stability, particularly in sunlight, and so on. In 1983, Philip Entwistle listed 31 Lepidopteran, 6 Hymenopteran, and 1 Coleopteran pest species for which control with baculoviruses has been demonstrated to be feasible or highly likely. There is a little or no current effort to apply viruses for control of disease vectors or stored product and timber pests. Due to their small size, symmetrical structural organization, load capacity, controllable self-assembly, and ease of modification, viruses can be used to act as nanoparticles that carry drugs or dyes, thus having therapeutic and imaging applications. They can also serve as advanced nanomaterial synthesis reactors. For instance, cowpea chlorotic mottle virus showed a highly dynamic platform with pH and metal ion-dependent structural transitions. In 1998, Douglas and Young in Montana State University made use of this capsid dynamics and exchanged their natural cargo, nucleic acid, with synthetic materials. At about the same time, the research team led by Mann, University of Bristol, used the rod-shaped particles of Tibetan mosaic virus as templates for the fabrication of a range of metallized nanotube structures using mineralization techniques. Following that invention, tobacco mosaic virus particles have been utilized to generate various structures, nanotubes and nanovirus for use in batteries and data storage devices. Rajan Muthuramalingam from University of Madras, India, showed that plant virus nanoparticles are not infectious toward mammalian cells. Plant viruses have a size particularly suitable for nanoscale applications and can offer several advantages, including biodegradability, variety of shapes and sizes, and ease of production. Viruses and their non-infectious counterparts can be produced through molecular farming in plants or fermentation in cell culture. As we could see today, modification and genetic engineering methods can be used to make modified genomes that can be carried into plants and animals by viruses, acting as vectors or vehicles, which we are going to review in our next lecture. Please feel free to leave your questions and comments in the forum, and we'll be happy to answer them. Good luck, and looking forward to seeing you soon. AJ.